Good morning. Today I'm going to talk about the renal system or the urinary system. The urinary system is consists of two kidneys, two ureters, and we have unpaired bladder and urethra. As we know, the kidneys is the site for the urine production. Then it will be transmitted through the ureters toward the bladder and from the bladder toward the urethra. The main function of the renal system, as I said, production and excretion of the urine. Furthermore, regulate electrolyte levels in the extracellular fluid. So if there is increase in the sodium level in the blood, through the urine, the body is going to excrete the excess of that sodium minerals. The other function we have hormone synthesis. We have two hormones that produce through the kidney or inside the kidney. We have the renin and we have the erythropoietin. Renin play a role in the regulation of the blood pressure, whereas the erythropoietin, from the name, control the development and trigger the development and formation of the red blood cells in the bone marrow. The typical shape of the kidney is the bean shape, and usually in the medial side we have medial indentation, which we call it the hilus. From outside, the kidney is surrounded by a capsule. This capsule consists of collagen fiber. This is a very strong capsule and prevent the expansion of the kidney during the inflammation. Sometimes this capsule may contain smooth muscles and also have a layers of dense irregular connective tissue. If we cut the kidney in longitudinal section, we have two portions. We have this portion that we found outside, this called the cortex, whereas the side we found here in the interior of the kidney is called the medulla. So we have the cortex and we have the medulla. Usually the cortex receive about 90% of the blood supply that goes to the kidney through the ureter. So this is the capsule. As I said, it's dense irregular connective tissue. Sometimes it may contain smooth muscles. As I said, this is very strong. This prevents the expansion of the kidney during the inflammation, and this will result in increased pressure in these tubules inside. The area just below the capsule is called the cortex, and it contains these tubules, all what we can see inside the kidney, these tubules, and also we have this structure. This is only found in the cortex. This one is called the renal corpuscles. The functional unit of the kidney, which is the smallest unit that can perform a function, we call it the uriniferous tubule. And usually this uriniferous tubule consists of two parts. We have the nephron and we have the collecting duct. The reason they divided into two portions, the nephron and the collecting duct, because the embryonic origin of the nephron is different from the embryonic origin of the collecting tubules. In a human, each kidney contains around 2 million of nephrons per kidney. Also in the animals, reach to the level of hundreds of thousands per kidney. As I said, the kidney receives 95% of the cardiac output. Every heartbeat, 25% of that output goes to the kidney, and 90% of that 25 goes to the cortex. As I said, this is the typical shape, which is the bean shape. We have medial indentation, which we call it the renal hilus. This area here just below the capsule, we call it the cortex, and the one that we have here, we have the area of the medulla. We can see here this structure here, we call it the medullary pyramids. Medullary pyramids. And usually this medullary pyramid with adjacent area from the cortex, we call it renal loop. So the renal loop is the renal pyramid and associated cortical tissue. So this is this is the area of the pyramid, and this is the associated part from the cortex. Both together we call them as the renal loop. As I said, the kidney we divide it into two portions. We have the cortex and we have the medulla. In the cortex, we have the area, we have these tubules, and very important, we have this structure here, which is the renal corpuscle. As we can see here, the area of the medulla, only we can see these tubules. This area here, the junction between the cortex and medulla, we call it the cortex medullary junction. As I said, we divide the kidney into two portions, cortex, medulla, and we can see here this is the area of the renal corpuscle that only found in the area of the cortex, and here we have the tubules. As I said, renal medulla is the area just below the cortex, and there's some extension from the medulla into the cortex. You see this is the area of the cortex, and you can see these straight tubules. See this one here. This extension from the medulla toward the cortex, we called it the medullary rays. And if we have some structure like this one here extend from the cortex toward the medulla, we call them as the renal columns. So this is one with higher magnification. See this area of the renal corpuscles, and we can see this extension. These are straight tubules that extend from the medulla toward the cortex. These are called the medullary rays. As I said before, kidney, the bean shape, the typical shape. Inside the medulla, we have these medullary pyramids with the associate cortical tissue. We call them as the renal loop. Usually, we can see here this kidney consists of several medullary pyramids with adjacent area from the cortex. All of these structure we call as the renal loop. And because this kidney have several loops, they call it the multi-lobar kidney. So in a human, for example, we have 8 to 10 renal pyramids with associate part from the cortex. We call it as a renal loop. So in this kidney, we have between 8 to 18 renal loops, and we can see each one is open differently into the urethra, like open separately. So this is the area. Again, this is the medullary pyramid. This is the adjacent area. We call it the renal loop, and we can see each loop opens separately into the urethra. As I said, the functional unit of the kidney is the nephron. As I said, the nephron, we'll talk about it later, the structure that is composed, but now we'll talk about the classification of the nephron. Usually the classification of the nephron depends on the location of the renal corpuscle. If they found in the upper portion near the capsule, we call them superficial cortical nephron. If we find them in the middle, we call them the mid-cortical nephron. If we find them near the junction between the cortex and medulla, we call them the juxtamedullary nephrons. So we'll talk about the structure of the nephrons. The nephron consists of the first part, which is the renal corpuscle. Then we have the proximal convoluted tubule. Then we have this tubule that's going to descend and then reverse in direction and going to ascend. This we call it the lobe of Henle. Okay, and the last portion, this one here, we call it as the distal convoluted tubules. And this distal convoluted tubules is going to open in the collecting duct. So these structure 1, 2, 3, 4, all of these are structures of the nephron. We have the renal corpuscles, we have the proximal convoluted tubules, we have the lobe of Henle, and we have the distal convoluted tubules. For the lobe of Henle, you can see here this portion is thick and this one is thin. We call this one thick portion of the lobe of Henle. And if it's going down, we call it the descending, and if it's going up, we call it as the ascending. So we'll start with the first part, which is the renal corpuscles. It consists of two parts. We have the glomerulus and we have the Bowman's capsule. The first part, which is the Bowman's capsule, which is the blind end tube, see like this. This is a tube, we call it the blind end because it have only one opening. And this area here, it will be like this, which is this one here. This area here we call it as the Bowman's capsule. This Bowman's capsule, we have two parts. The one that found outside, we call it as the parietal layer, and it composed of simple squamous epithelium, and usually the lining, as I said, simple squamous, and it's the parietal layer. And we have the layer that found inside here and surrounded by the blood capillaries. These blood capillaries, we call it as the visceral layer. And again, this one is modified simple squamous epithelium. And the name of the cell, we call them as podocyte. Podocyte means that they have legs, because these cells, I will show you later, almost like they have legs. Okay, so that's why they call it as the podocyte. So the first structure of the renal corpuscle, which is the Bowman's capsule, which blind end tube, as I said, it's invaginated blood vessels, which is the blood capillaries, which is the glomerulus, consists of two layers. The one that found inside near the blood capillaries, we call it as the visceral layer, composed simple squamous epithelium. These cells are modified because they have legs. They call them as podocyte. And the one that found outside, they call it as the parietal layer. So this is the first structure here, which is the Bowman's capsule, and it composed, as I said, from the blind end tube, and inside it we have the blood capillary, which are the glomerulus. As I said, these cells, which is the visceral layer, because they have modified long cytoplasmic processes, see this one here, almost like they call them as podocyte. And these podocytes compose of two parts. We have the big portion here, we call them as the primary processes, whereas the smaller one, we call them as the secondary processes. Sometimes we call them as pedicels. Okay, as I said, these are the visceral layer of the Bowman's capsule. Very important, you can see here that the secondary process interdigitate like this, like finger inside each other, and there will be like a spaces formed. This space formed here, we call it as filtration slits. And this very important because the filtration of the blood start from this area. So we have the podocyte. The podocyte is the modified squamous epithelium of the visceral layers. As I said, they have primary processes and they have secondary processes. The interdigitation lead to formation of the space. This space, like this one we can see here, this called as the filtration slits. So this one, the simple squamous epithelium. This is the primary processes, and this one we can see it here down there. This is the secondary, and the space is formed near because of the interdigitation of the secondary processes, the pedicel, we call it as the filtration slits. These cells, the simple squamous epithelium, usually they lie into a basement membrane, and usually in this basement membrane, to ensure the filtration, we have fenestration, and this fenestration also extend toward the endothelial cells of the blood glomerulus. And this is very important because we need the small particles to be filtered from the blood, whereas the large particle should be stopped from filtration. This is the blood capillaries, and we can see here this is the area of the simple squamous, and this is the parietal layer, and here the area where the urine will be collected. This one here we have the parietal layer of the Bowman's capsule, whereas the one that surrounds the blood capillaries we call it as the visceral layer. So again, this is the epithelium with the long primary processes, and we have the secondary, all of these secondary processes, and the space that form between them, this is the filtration slits. And we can see here this is the basement membrane, and inside here we have the endothelial with the fenestration, as I said, to allow the small particles to be filtered, whereas the large particles will not be allowed to pass through this fenestration. Again, this is the pedicels, which is the foot processes. This is the cells, and we have these are the primary, and we can see the smaller one, the secondary. And we can see here these spaces here, these are the filtration slits. And this structure we can see here, this is the area where there's a filtration of the blood and start to form the urine. The second structure we have in the renal corpuscles, the renal glomerulus. Very simple, it consists of blood capillary. This is just blood capillaries, and these blood capillaries is lined by fenestrated endothelial cells. And we can see here this is the area of the basement membrane. As we can see here, this basement membrane is a fused basement membrane of the simple squamous of the podocyte and the simple squamous of the endothelial cells. So this is the area, this is the area here, this is the area of the parietal layer. Inside here we have the visceral layer. As we can see here, this is the area where the urine will be collected, so that's why we call it the urinary pool. And this here This is the area where the blood vessels that going to enter the kidney and leave as the efferent and efferent arteriole. So this area here we call it as the vascular pool. The space that found between the visceral layer and the parietal layer is called the Bowman's space or the urinary space, whereas the area just opposite to the vascular pool, which would be at this area here, we call it as the urinary pool. And from this area here, this will lead to the proximal convoluted tubules. So again, this is the Bowman's capsule with the Bowman's space. This is the whole structure here. This is the renal corpuscle. From outside we have the parietal layer. Inside here we have the visceral layer of the Bowman's capsule. As I said, this is the area of the blood vessels. This area here we call as the vascular pool, whereas this area here we call it as the urinary pool. And this area where the urine will be collected after the filtration process, we call it as the urinary space. So we'll talk about the renal filtration barrier. As we know that there will be filtration of the blood, and then will be reabsorption process to ensure that only the wasted material to be excreted, whereas the useful material to be reabsorbed. So usually for the renal filtration barrier we have glomerulus, capillary, fenestrated. We have the endothelial and basal lamina and filtration slits with diaphragm that found between the pedicels. Just go back here. Just to remind you here, this is the endothelial cell, and you can see here the fenestration, and then we have the fused basement membrane for both the endothelium and the podocyte. And at this area here we have the filtration slits as they form because of the interdigitation of the secondary processes. This structure here we call it as the blood urine barrier. Okay, this is the separation between the blood and the urine. The separation is formed from the endothelium, the fenestrated endothelium, the fused base membrane. Again, there is a fenestration, and then we have the podocyte. From the podocyte, we have the secondary pedicles that interdigitate and form the filtration slits. As we said, that there's like a diaphragm that control the fluid to pass through these filtration slits. These filtration barrier very important because they allow water, ion, and small molecules to pass from the blood to the urine space, and very important prevent the passage of large or negatively charged protein. Very important, the urine is the ultrafiltrate of the blood plasma. The second structure we have after the renal corpuscle, we have the proximal convoluted tubules, and usually the drain of the Bowman's space at the urinary pool toward the proximal convoluted tubules. The lining, simple cuboidal epithelium, and this epithelium they have microvilli. So the lining, simple cuboidal epithelium with the microvillus brush border. The function of the proximal convoluted tubule to reabsorb of all glucose, amino acid, small proteins. All of these structures are useful material, and the body need to keep them. See this one here, also sodium chloride and the water. all of these structures have to be reabsorbed. So this area here, this is the area of the vascular pool, and this is the urinary pool, and this structure here, all of the urine is going from the area of the Bowman's space or urinary space toward the proximal convoluted tubules. As I said, simple cuboidal epithelium with brush border or microvillus. We can see here most of the structures of the proximal convoluted tubule. We can see them in the area of the cortex. The lining simple cuboidal with a brush border. All of these structures are the proximal convoluted tubules. Also, we can see the distal convoluted tubules, but notice the difference here, that the profile of the proximal convoluted tubules is more than the distal convoluted tubules. The reason for that, because the proximal convoluted is longer than the distal convoluted tubules. The other thing, see the lumen here? We can see a clear lumen, whereas here we can see all very limited space. Here because we have the microvilli, and here we don't have the microvilli. So this is the important difference between the proximal convoluted and the distal convoluted tubules. After that, we have the descending or the lobe of Henle, and as I said, lobe of Henle consists of two portions. We have the descending part and we have the ascending part. The second thing we have this portion here, we call it as the thick portion, whereas this one here we call it as the thin portion. They call it descending thick portion because the lining is simple cuboidal epithelium with a prominent brush border, and usually this structure we can see in the area of the medullary rays. See this one here, all of these structures is the thick portion of lobe of Henle. Whereas when this epithelium is changed into simple squamous epithelium, we call it as the thin limb of Henle's lobe. As I said, the lining will be simple squamous epithelium. See here, this is the epithelium, which is the simple cuboidal epithelium. This is the thick portion. However, if we go to this portion here, we call it as the thin portion. Then we have the ascending portion. Again, it consists of two portions. We have the thin portion, or call it as the ascending thin portion, and the second part we call it as the ascending thick limb of Henle's lobe. Again, the lining starts with simple squamous epithelium, whereas this one here is cuboidal epithelium. Then we have the ascending thick limb of Henle's lobe, and this one here so we reach to the level of the Henle's lobe. As I said, we have two portions. We have the descending portion and we have the ascending portion. And the descending portion and ascending portion, both of them divide into two portions. We have the portion that have the lining, simple cuboidal epithelium, and then we have the lining, which is the simple squamous epithelium. Then again, simple cuboidal epithelium. This portion here we call it as the thick portion, and here we call it as the thin portion. And we have descending thick portion, and we have descending thin portion, and we have the ascending thin portion, and we have the ascending thick portion. The only differentiation between them, to differentiate between this the thick or the thin, is to observe the epithelium lining. If it's cuboidal, then it's the thick portion, and if it's squamous, then it will be the thin portion. As we can see here, this is the area of the epithelium, and this is again the epithelium, simple cuboidal, whereas at this area here, the epithelium is changed into simple squamous epithelium. So this is the thin portion. Again, this is the epithelium, simple squamous, whereas the epithelium at this side here, we have the cuboidal epithelium. This is the thick portion, whereas the one that we have here, this is the thin portion of Henle's loop. As I said, we have the descending and then we have the ascending portion. For the ascending thick limb of Henle's loop, also we call it as the straight portion. This one here, the tubes start to be straight, simple cuboidal epithelium, then start to convoluted, and then we'll call it as the distal convoluted epithelium. Usually the lining of this epithelium or this tubules, simple cuboidal epithelium, there's no brush border. See this one here? This is the ascending thick portion. The lining epithelium, which is simple cuboidal epithelium. As I said, there is no brush border. There's no microvilli. At this area here, near the vascular pool of the renal corpuscle, we can see this epithelium. If instead to be simple cuboidal epithelium, they almost condense near each other, and the cells are columnar. This portion here, this one here, we call it as the macula densa, and this part is part from the juxtaglomerular apparatus that play a role in the regulation of the blood pressure. Then we have this convoluted tubules, which is similar to the histologically to the ascending thick portion of Henle's loop. The epithelium, simple cuboidal epithelium, they have a wider lumen than the proximal convoluted tubules, and very important, they lack the brush border. So the first point, the lumen is wider, and the second point is they lack the brush border. The main function again to reabsorb the sodium ion from the filtrate. The last portion from the proximal convoluted or from the nephron, see this area here? This area here we call it as the connecting tubules. From the name, they connect the nephron with the collecting duct. And usually the lining is simple cuboidal epithelium. We have two types of the cuboidal epithelium. We have the principal cells and we have the intercalated cells. The principal cells, which is the light cells, whereas the intercalated are the dark cells. These cells will be lying in the basement membrane. These cells are very important in removing the sodium ion from the filtrate and secrete the potassium into the filtrate or in the urine, whereas the intercalated, they have many apical vesicles or mitochondria, and the main function remove of the potassium from the filtrate and secrete water. So the principal cells will be responsible for the reabsorption of the sodium and secrete potassium, whereas the intercalated will be responsible for reabsorption of the potassium and release of the water. So this is the connecting tubule. Again, the epithelium lining is simple cuboidal epithelium. The last portion of the urinary nephros tubules, as I said, we have nephron and we have the collecting tubules. The collecting tubules, their segment mostly found in the medullary rays, and we have two portions. We have the cortical portion and we have the medullary portion, and we can see it's open into the urethra. It's open to the ureters. As I said, it consists of two portions. We have the cortical portion and we have the medullary portion. Again, we have two types of the epithelium, which are the simple cuboidal epithelium. We have the principal cells and we have the intercalated cells. The principal cells, we can see here, there's a xylem, whereas the cells that have apical vesicles, we call it as the intercalated disc. For these two types of cell, we could not differentiate under regular microscope. We need to use the electron microscope. As I said, for the collecting tubules, we have the medullary cortical portion, and the last portion that found in the medulla, we call it again as the medullary portion. And again, they have both type of cells, the principal cells and the intercalated cells. This slide here, this is the area of the cortex, and we can see here these tubules. We have the distal convoluted and we have the proximal. See here, the lumen is wider compared to this one here, and here we could not see the brush border, which is the landmark for the proximal convoluted tubules. If we look at the collecting tubules that found in the medulla, you can see these here. These are the simple cuboidal epithelium. This is the simple cuboidal epithelium, and you can see here, we can see squamous epithelium. Then we look at the thin of lobe of Henle. Now we are going to describe the blood supply of the kidney. The blood supply of the kidney is through the renal artery. This renal artery, once he enters the kidney, start to divide, to branch. Each one of these are called the interlobar artery. Interlobar artery, and usually this interlobar artery set the boundaries for these medullary pyramids. So it's very important because it divides the kidneys into renal lobes. Then this artery, at the level of the junction between the cortex and medulla, change the shape like arc, and they call it the arcuate artery. And from this arcuate artery, we have branches. These branches are called the interlobular arteries. So we have the renal artery, once entered through the hilus, give it branches, the interlobar arteries, then at the level of the junction between the cortex and medulla, the name is changed into the arcuate artery, and from the arcuate artery we have the interlobar arteriole. So we reach to this level here, and all of these are the interlobar. Then the interlobar will give us a branch, which is the efferent arteriole, and this efferent arteriole will give us the capillaries that going to go inside the renal corpuscles and exit as the efferent arteriole. And from this efferent arteriole, There will be blood capillaries, and then we go to the level of the post-capillary venules. See this one here? It's the interloper, and from this one is the afferent arteriole, and it's going inside the renal corpuscle to give us the glomerulus. Very important. Typical circulation, we have arteriole, capillary, venules. However, in the kidney, we have the afferent arteriole, we have the capillaries of the glomerulus in the renal corpuscle, then we have afferent arteriole, then another capillaries is formed from the afferent arteriole, and this afferent arteriole capillary then goes to the venules. So instead going directly from arteriole, capillary venule is going afferent arterioles, capillary, afferent arterioles, then capillary, and finally we have the venules. After that we have the ureter, which is muscular tube, and usually conduct urine to the bladder from the renal pelvis. The control of the ureter, again because the wall contains smooth muscles, will be through the sympathetic and the parasympathetic innervation. For the lining epithelium, usually the epithelium is transitional epithelium. The upper two-thirds has two layers of smooth muscles. We have the inner longitudinal and we have the outer circular smooth muscle. However, the distal one-third, they have additional outer longitudinal layer of smooth muscle. So here we have, instead of two layers, we have three layers compared with this one here. We have two layers. However, the distal portion have three layers. They have the additional longitudinal layer, which is the inner layer. So this is the epithelium. This epithelium is transitional epithelium. And this is the area of the lamina propria with some mucosa. And this is the area of the muscles. We have the inner longitudinal and we have the outer circular. However, in the distal portion, we have this epithelium here. We have three layers. We have the inner longitudinal, we have the middle circular, and we have the outer longitudinal. The organ after the ureter, we have the urinary bladder. The epithelium is transitional epithelium. This epithelium is pseudostratified columnar epithelium, but because it's transit between two stages, they call it as transitional epithelium. They found when the bladder is relaxed, the bladder is empty, it appears as stratified squamous epithelium. However, when the bladder is full of urine, the epithelium appears as one layer, which is the simple squamous epithelium. So that's why they call it transitional epithelium, because it's a transit between two stages, relaxed or distended. So we can see here this epithelium, stratified epithelium. It's a type from the pseudostratified epithelium. See here, this is the epithelium. This is the epithelium. And this portion here, this is the upper portion cells. And these cells, they are wider than they are tall, which is not a characteristic from any type of epithelium we studied before. We remember squamous epithelium, they are flattened, cuboidal, they are tall, equal the width, columnar, they are taller than the width. However, in these cells, they are wider than they are tall. These cells is called pillow cells. As I said, when the bladder is distended, full with urine, the epithelium appear as squamous epithelium. The last organ that I'm going to talk about in this lecture, which is the urethra, which is the part from the male and female renal system. In the male also is part from the male reproductive system. Again, in the male, it carries the urine and also carries the semen. It have two layers of smooth muscle. We have inner longitudinal and outer circular, almost like the urethra. In the male urethra is divided into three portions. We have prostatic urethra, which is near, this is the bladder, and near the neck of the bladder, there is the prostatic gland. This portion called the prostatic urethra. Then we have the portion that found inside the pelvic region. We call it the membranous. And finally, the one that found inside the penis, we call it as the penile urethra. For the prostatic urethra, the epithelium is transitional epithelium, similar to the urethra and bladder. Then we have the membranous and penile portion. The epithelium is pseudostratified epithelium or sometimes stratified columnar epithelium. Near the opening, the epithelium is changed into stratified squamous epithelium. This is the area of the urethra, inside the penis, and outside we can see all of these structures, which is the area of the prostate. So this portion of the urethra we call it as the prostatic urethra. As I said, the end portion of the urethra is lined by stratified squamous epithelium. In the female, the urethra is short compared to the male. It's around 3 to 5 centimeters in length, lined primarily by stratified epithelium, which is different from the male. As I said, the first portion is transitional epithelium, then changed to the pseudostratified, then stratified columnar epithelium. However, in the female, it's surrounded by or lined by stratified squamous epithelium. However, maybe some areas of the urethra, the epithelium have pseudostratified columnar epithelium, also sometimes stratified squamous epithelium.